Check on YouTube.
सब बेटा लगे नहीं थे
YouTube check कर.
Good evening, sir.
Very good evening. जोर से कहो किसी को. कहाँ है? कहो. Okay. YouTube আছে. YouTube আছে. खाना जी नहीं देनी.
Good evening, sir.
Very good evening. Okay. So good evening all. Okay. So let's wait for a couple of minutes. So screen is visible to all of you.
Yes, sir.
Okay. So, so all of you aware of the topic, today's topic. So what topic we will go for?
Yes, sir.
Sir.
Okay, fine, very good,
Biochemistry.
very good, very good.
Biochemistry.
Okay.
Biochemistry.
Yes, yes, yes. So biochemistry is a kind of your easy subject or hard subject? Arey, tell me, share your views regarding biochemistry.
Sir, moderate.
Moderate. Okay, good. Then anybody else who thought that it's a kind of your hard subject? Anybody? Feel free to share your views. Okay, okay, fine. Yeah, okay. So without any delay, we'll start the session. So some students are still joining. Very good. Okay, fine. Very good. First Mr. Topic. Okay. But irrespective of your whatever kind of semester, it's a kind of your crucial subject for your entrance examination. Okay, important subjects. Okay, so irrespective of your hardness and your easiness, we will make it uh, as a kind of your fun. Okay, so that we can memorize the bio and understand the biochemistry, okay, easily. Very good. Okay, fine. Yeah. Okay, most of you have already studied this kind of topic, but I need to recall or revise that kind of thing. Okay, what kind of question also there? Okay, so main topic you need to go for it. Okay, so without any delay, it's 7 8. Okay, so we will start. Manisha. Those once I will start, those will join after that. They won't get any kind of, they couldn't cannot understand. Okay, so okay, so we'll start. Fine. Yeah. Is it visible to all of you? দেখা যাଉଛି screen? Okay. So the
Yes,
main
sir.
Yes,
thing.
sir. Yes, sir.
Okay. I got it. Okay. Yeah. So so on the main that will be biochemistry. So we'll go for the amino acids. Okay. Or amino acids. What is the amino acids? Anybody? What are amino acids? କିଏ କହିପାରିବ?
Sir, composed of a carbon group.
Okay. Composed of
Building
carbon.
blocks of protein.
Very good. Building blocks of protein. Okay.
Proteins.
Or we can call it
Monomeric
as.
units of protein.
Very good. Very good. Yeah. So it's a kind of your monomer of your protein. Okay. Or you can, according to you, that will be your building blocks of your protein. Okay. So now we will go. Before going to your amino acids, you will know it's a kind of your biomolecules. Okay. So before going to your amino acids, we will go for, we need to know what is your biomolecules. Okay. So, okay. So these are the biologically active molecules, okay, which are responsible for the living organism, for the growth, sustenance, as well as biological process of the living organism. Okay. So mainly these are the mostly biologically active molecule. Okay, so biomolecules, for example, your protein, lipids, carbs. Okay, then your nucleic acids. Okay, so nucleic acids may be DNA or RNA. Okay, so these are the biomolecules. Okay, so biomolecules, what are the main atoms? Molecules are also there in the biomolecules. So mostly chunks. Okay, it's a kind of your you can memorize the components. Okay, that will be your chunks. CHONKS, chunks. Okay, so C stands for carbon, hydrogen, oxygen, nitrogen, potassium, sulfur. Okay, ବୁଝିହେଲା? chunks. chunks ମାନେ ମନେ ରଖିପାରିବ?
Yes, sir.
So that will be from the gradation. So which one is the
Yes,
first
sir.
କୋଉଟା ଅଛି?
Yes, sir.
First କୋଉଟା ଅଛି ରେ? Carbon. So it is the main component.
Carbon.
Very good. Okay. So if you look,
Carbon.
when we consider one living organism, okay, the total, it's a kind of your 50% of your biomass. Biomass mane, that is the biomass is the dry weight of the cell or organism. Okay, so what is dry weight? Okay, so we need to ame ka karma gute the organism ke we will put into an oven. Okay, at an temperature of 60 degree for 24 hours. After that we will take that organism weight, and that will be your biomass. So without devoid of any kind of your water molecule or water content, that will be your biomass. So from the biomass of an organism or cell, so the carbon content is more than 50%. Clear? Why it is important for the living organism? There is a catenation. What is catenation? So carbon can mix. Okay, so this kind of thing is catenation. Whether in the linear chain or it can form the ring compound. Ring compound is known as aromatic compound, and chain is the aliphatic compound. So it will adjust in both aliphatic and aromatic compound. Clear? So this is the biomolecules. Okay. So as I have already told you, what are the kind of your biomolecules? That will be carbohydrate, fatty acids, amino acids, nucleotides. Okay, so that will be first one sugar. So that is the monomer of your polysaccharide. Okay, then fatty acid, monomer of lipids and fats. Amino acid we have already discussed. That is the proteins. Okay, तापर है nucleotides. What is nucleotides? किए कहि पारबो? What is nucleotides?
Nitrogenous bases plus ribosomal plus phosphate group make nucleotides.
Very good. Nitrogenous base and base, then pentose sugar. Okay, then it will be, you will get, I'm writing here U4. Okay, so this is the nucleotide. So do you know what is the difference between nucleotide and nucleoside? ଜାଣିଛ ରେ କୁହ
Yes, sir.
କୁହ କିଏ କହିପାରିବ?
Yes. Nucleoside is different from phosphate.
Very good. If we cut down the phosphate group, then it will be your nucleoside. Okay. So nucleotides are the monomer of your nucleic acid. Okay, clear? So now we will look into the your amino acids. Okay. So before going to this amino acids, we will know what are the main component of your or main groups are there in the amino acids. Okay. So it's a kind of your, as you can see, this is the common basic structure of your amino acids. Okay. So one carboxylic group is there, one amino group is there, one carboxy terminal and another your amino terminal is there. So anybody, can you tell me what is chiral carbon or chiral carbon? Okay, got it, very good, got it. Okay, yeah. So chiral carbon is the, if one carbon atom is attached to the 4 different kinds of group, that will be your chiral carbon. Okay. So what is alpha carbon? Anybody? Alpha. Plus 2 रे पढ़ी जो समझते को?
Linked to the carbon which is attached to the functional group.
The carbon which is attached to the functional group.
Carbon closest to the functional group.
Very good, very good. So this carbon that will be attached to one, this is the functional group. Okay, this is here. So which one is the alpha carbon? It is alpha carbon, right? ठीक अच्छे? ठीक अच्छे कि नहीं रहे?
Yes, sir.
Okay, very good. Okay.
Yes, sir.
Okay. So basically in the amino acid we have one amino group and one carboxylic group. Okay. So as you can see here, so these are consisting of your amino group NH2 and another one is your carboxylic group COOH. Okay. So in the one direction you will get the amino end or amino terminal or N terminal. In another hand you will get the carboxy terminal or carboxyl group, okay, or C terminal you can say. Okay, so you can say the two in the protein, the two amino acids are joined together by which kind of interactions? Which kind of interactions? কে কই পারবো? Two amino acids are joined by means of which force of interaction? Huh? Okay, very good. Peptide bond. Okay.
Peptide bonds.
देखी पकालो ना रे. हाँ देखी की कोई दिया लिखा है. हाँ very good. Very good, very good. Okay. So देखो. Now question is तो लिखा ही है ना. So how the question will be in the Cpet question examination. So तुम्हारे पास देखो you have the answer. Now I am asking. Okay. So the peptide bond, बहुत से ध्यान से सुनो. The peptide bond in the amino between the amino acids, it is option दूची देखो. Sorry, जिन पेज दूची से तेरे दूची. Option 1 single bond, option 2 double bond, option 3 triple bond, option 4 none of the above. Nota. Nota कहीं दबारे. Let it be partial double bond. So the peptide bond is which kind of bond? Whether it is single bond, triple bond, or partial double bond.
Partial double bond.
So what is the what kind of
Partial double bond.
Very good. Okay. So how it will be partial double bond? Why not it is fully double bond? Oh, okay. So if you have the question like this, option 1 single bond, option 2 double bond, option 3
Because the end
okay, wait. I will ask you. Okay. So option 1 single bond, option 2 double bond, option 3 triple bond, option 3 none of the above or all of the above. This kind of thing will be there. So then we will go for option 2, that will be double bond. If partial word will be there, okay, single bond, double bond, partial double bond, partial single bond, or that will be your partial double bond. Clear?
Yes, sir.
Okay,
Yes,
fine.
sir.
Yeah.
Yes, sir.
So how it's a kind of your mechanism, how your double bond is there formation, how peptide bond formation will be there, how water molecule will be liberated to give rise to your peptide bond. Okay, this kind of thing we will study in the later classes. Okay. Okay. So if for example protein ray, if amino acid conformation will be changed, then the protein's functional variation will be there. For example, sickle cell anemia रे. Sickle cell anemia रे कौन हो रे? Why sickle cell anemia will be there?
Sir, RBC structure change है.
हाँ, RBC structure काहे की change हुए? Which particular thing is responsible for change in the RBC structure? Okay, very good. So, हाँ,
Glutamic acid valine कौन है?
So single change in single amino acid it changes. That will be glutamine to valine. We will look into the structure of glutamine, valine. Okay. So the structure of protein is changed or functional deformity will be there due to change in the amino. Okay, fine. Now it's a kind of your data.
Yes, sir.
Okay.
Yes,
so,
sir.
okay. Now it's a kind of your data. So just remember the smallest peptide, that is the glutathione. Okay, so glutathione, you have 3 amino acids. Okay, so glutathione, it's a kind of your peptide. Okay, smallest peptide, that is consisting of your 3 amino acids. So do you know what is antioxidant? Antioxidant जानते हो क्या ना रे? एंटीऑक्सीडेंट काना जानी छो कि नहीं?
नहीं सर।
नहीं?
यस सर।
ग्रीन टी पीने के स्लिम हवा लगी। अरे अरे ग्रीन टी पी ले ऐड दस
मैन-मेड और नेचुरल substances that may prevent some biological damage.
अरे ग्रीन टी रे जो ऐड दिए माने स्लिम हे जीवो ये हे जीवो सब हे जीवो कोई का ठीक ना बोल? हां
Yes sir. Yes sir.
नहीं सते पतला हो जंती। अरे ମୁଁ ତ ବର୍ଷ ବର୍ଷ ହେଲା ପିଉଛି ପତଲା ହଉନି ଯେ।
सर এটা
रेगुलर अरे ग्रीन टी जदि पहाड़ के जाएगी ना तो मोस्टली ग्रीन टी इज नॉट रेस्पॉन्सिबल फॉर योर पतला और मोटा बुझेला। सो दैट विल बी रेस्पॉन्सिबल फॉर योर दैट विल बी योर व्हाट दैट विल बी metabolism will be high. Okay, metabolism will be in the good way. Okay, so if metabolism will be there and good, then you will get the good digestion. Okay, then you will get the no toxic material deposited in your body. Okay, that can be removed. Square bracketed कौन रे 6 सेमिस्टर रे अच्छे तुम ROS reactive oxygen species इटा काना किए कोई परबो? अरे oxygen within square bracket, what is this?
Nascent oxygen.
Very good, nascent oxygen. So antioxidant is the molecule which can destabilize this kind of thing. Okay, so this nascent oxygen is highly reactive. That can bind to your DNA, proteins, okay, lipid due to this. जेंटा कि तुम्हार OH minus, O3 minus, O2 minus, these are the free radicals. They are the highly reactive species. Okay, next you will see titin, one largest polypeptide that is consisting of your 34,350 amino acids. So you will get mostly in the muscles. Okay, so there your contraction and relaxation, that will be your muscles elasticity is regulated by this kind of thing, this kind of protein. It is a titin. So that is the largest peptide. Okay, till today. Clear? So apart from this, the amino acids and proteins are helpful for in the enzyme synthesis, hormones, transporters, antibodies. Okay, so how many number of your amino acids are there? Standard amino acids? 20. So here written 22. Okay, so 20 standard amino acids are there. Apart from 20 you will get another two. One is your selenocysteine, another one is your pyrolysine. Okay, so if you a question asked to get dhyan, first one amino acid that is discovered, that is the asparagine. Clear? Okay, and last one amino acid that is discovered, this is the threonine. Okay, so from 1806 to 1938. Bujiela? Okay. First amino acid synthesized in our body, that is the methionine. Okay, so now tell me, but in case of your prokaryote and eukaryote, you will get a kind of your different thing. Okay, in both case you will get the methionine as the first amino acid, but in case of your prokaryote you get fmet. Fmet means that is a formilated methionine in case of your prokaryote, but in case of eukaryote you will get simple methionine. Clear? Bujiala? Okay, so apart from your common structure of your amino acid, that will be your one amino terminal, one carboxy terminal, one carbon, and another one is your R group. So if you change in the R, we will get different kind of amino acids. Okay, clear? Bujela. So now we will see the different kinds of amino acids, and I will I will let you know how to remember these all the kind of your amino acids. Okay, तो आज क्लास रे ही तुमको पूरा मन रखे देबी पूरा टोटल एमिनो एसिड्स हे परबा कि नहीं पॉसिबल इज इट पॉसिबल
Yes, sir.
yes
Yes, sir.
very good
Yes, sir.
okay etki gods थिवर कथा very good okay so first हमें देखि दो हला so what is the basic structure of your amino acid oxy terminal this is the common for all okay this is common for all except proline okay देखो काना अछि
Zoom करो.
okay fine
Sir.
एबे असला एबे क्लियर जूम कर देछि आउ जूम
Sir, thank you. ना ना ठीक अछि.
ओके ओके फाइन एबे देखा जाउछि फर्स्ट हमें देखिबा सिंपलेस्ट एमिनो एसिड एमिनो टर ऑक्सी टर्मिनल ऑन इज योर आर जगह रे कोन लागि छि को तो आर जगह रे हाइड्रोजन लागला so that will be your glycine okay so in the three letter we can say it as gly okay that will be denoted by g okay next alanine h जगह रे ch3 okay r जगह रे तुम put it ch3 valine v then cysteine okay cysteine ch2sh just remember cysteine and your methionine these are the two amino acid which contain sulfur cysteine and methionine are the two Cpet report what is the write the two amino acid which chatta option de dithbo okay which of the following have the sulfur contain non polar amino acid okay to dekho tapore proline same ns2 n plus s3 then ch c double bond ch2 that is connected to your what amino group this kind of thing you will get only in the proline बुझियला बाकी सबुथरे r group is free next देखो next leucine ch2 ch देखो this it's a kind of modification of कोटा कहि परबो leucine is the modification of just simple modification of when you add in the valine one another carb ch2 that will be your leucine बुझियला मन रखा देखो valine the ch3 adding simple ch2 that will be leucine modification को तो car modification say same valine रो modification गुटे एंड रे आ गुटे तमे बढ़े दबो चेन बढ़े दलो ch3 ग्रुप टे लगे हां valine they just तुम सॉरी leucine रे गुटे सिंगल कार्बन टे लगे दलो that will be isoleucine next one is your methionine मन रखि दबो ए क्वेश्चन बार बार Cpet रे आछे which one is the sulfur containing cysteine and methionine which one is the first amino acid synthesized in the cell first amino acid methionine on in case of your prokaryote prokaryote F-met that is a formilated methionine very good okay next you will see the tryptophan okay tryptophan OH serine S then that will be CH OH CH3 okay that will be 309 T tyrosine CH one ring compound benzene ring and that is conjugated with OH 309 sorry tyrosine then asparagine ASN that is denoted by N CH2 C NS2 C double bond okay then glutamine CH2 C double bond why mine MINE will be there you need to have one amino group should be there NS2 group should be there बुझियला तापर देखो just positive, another two kinds of amino acid, positive amino acid, negative amino acid. Positive रे आसला तमर लाइसिन, आर्जिनिन, हिस्टिडिन. तमर स्ट्रक्चर पड़िबनी हला. जस्ट रिमेंबर positive amino acid. लाइसिन, K, आर्जिनिन, R, हिस्टिडिन. कौन? Histidine is denoted by what?
H.
H. Next one will be negative amino acid.
H.
Very good. Negative amino acid. Negative amino acids are glutamic acid or aspartic acid. Clear? Aspartic acid and glutamic acid. Okay. Don't be confused in glutamic acid, sorry, and glutamate. Aspartic acid, aspartate. Same genes or na different? Pyruvic acid, pyruvate.
Same.
Same. You can say it's same, but one hides the
Same, sir.
change होते हैं.
Which one is the protonated form and which one is the deprotonated form? Aspartic acid, sim add कर दो, that will be aspartate. Clear?
Aspartic acid.
Go. Just remember,
Yes, sir.
negative amino acid एते खाली क्वेश्चन रे पड़िबो, बेसी ये करबार मुंडो ये करबार नहीं हला. Aspartic acid, that is denoted by D, and glutamic acid, that will be denoted by E. Okay, so now we will memorize. Okay, so which one is the which kind of amino acid? Okay, देखा जाउछि रे समस्त को. अरे, this is the trick to remember your all amino acids. L stands for, किए कोई परबो L? L stands for कौन? अरे, L टा कौन को? Leucine. A, leucine. A stands for A टा कौन?
Alanine.
G लिखा है GLY. कौन हुए? GLY रे?
Glycine.
Very good. Very good. Then VAL टा कौन था?
Glycine.
VAL टा कौन?
Valine.
Valine. Very good. I टा कौन को? Iso लिखा है
Isoleucine.
Isoleucine.
P माने कौन? P PRO. Proline.
P stands for phenylalanine.
P proline. Phenylalanine होची F. ओके, देखो तो हमें केमती मन रखिबा MLAG VIP. MLA माने कना? Methionine, leucine. G GI लिखिची, G for glycine. VIP valine, isoleucine, proline. मन रहला non polar रो aliphatic मन रहला रे aliphatic और aromatic. हां, अरे डोंट वरी एते ये हो नहीं, टेंशन नहीं होनी. मन रहला कि नहीं? What is the difference between aliphatic and aromatic? जोटा तमर चेन कंपाउंड थिबो,
Aliphatic is chain.
linear compound, that will be aliphatic. Aromatic ring compound. Okay,
Ring compound. Aromatic.
very good. Okay, so MLHG VIP मन रहला aliphatic. बिना देखि कहि देबो aliphatic. जदि तुमको which one of the following amino acid is aliphatic हला देइतो मिथियोन एठु गुटे उठे के देतो मिथियोनाइन, phenylalanine देइतो serine, aspartic acid, histidine. कोउटा हबो? aliphatic amino acid कहलो serine दला, aspartic acid दला, histidine दला.
Methionine.
तो तमे जदि जानि थिबो दिस
Aliphatic.
MLAG VIP जदि मने रखिचो, then you can say easily कोइ परबो, मने रखिबार दरकार नै एते हला. GI खाली लिखाइ छि, खाली that will be glycine. Clear? मने रहला aliphatic amino acid.
Yes.
आ तुमको एबे तमर Cpet रे स्ट्रक्चर करो बोली कहिबोनी. हला जेतले एमएससी करिवो से जगह तुम करिवो पर कथा सेटा हमें एबे मने रखिबार अछि को जिंस कम टाइम भितर केमती मने रहबो हला एमएलजी वीआईपी एलिफेटिक देन एरोमेटिक कोन के कोइ परबो ट्रिप्टोफैन वाईटा कोन के कोइ परबो रे वाईटा कोन ट्रिप्टोफैन नॉट ट्रिप्टोफैन टायरोसिन ओके एबे देखो एफ स्टैंड्स फॉर फिनाइल एलानाइन देखो फिनाइल एलानाइन भी पी रे स्टार्ट हिची आ प्रोलीन भी पी रे स्टार्ट हिची हला जेतु फर्स्ट में प्रोलीन को पी दे देला तो फिनाइल एलानाइन को पी तो देई हबोनी ना तो फिनाइल एलानाइन केना जानिबो केमती साउंड होची फी रे को लेटर आछी फी रे को लेटर आछी फी साउंड रे को लेटर होची फी एफ हला सो ताकू एफ दिया गला बुझी हला तो डोंट बी कंफ्यूज इन प्रोलीन एंड फिनाइल एलानाइन बोथ आर स्टार्टिंग फ्रॉम पी बट जेतु हमें पी प्रोलीन को पी दे देला फिनाइल एलानाइन फी साउंड होची so that will be f starting from f okay wife टा कोन एरोमेटिक केमते मन रखिबो सर हैविंग परफ्यूम टा कोन मन रहला रे
Yes, sir.
कार मन नै रहला को
Yes, sir.
थैंक यू हमें यू न लिखी कौन लग कला कौन कला रे क्यू लिखिला ला एस स्टैंड्स फॉर सेरीन सी स्टैंड्स फॉर के कोई परबो CYS सिस्टीन ASN एन कण कोन को आस्पार्जिन यू बुझेला सचिन थैंक